Got it. Okay. So yeah, it's going to be a bit different, more on the computational side, but hopefully you'll see the value. Okay. So transcriptome variations. There's two main ways that people usually think about transcriptome variations. And I'll also connect that to something called Sapir-Whorf hypothesis. Anybody knows what it is before you look at your cell phones and look it up? Okay, so yeah, we'll get back to what that is and how's that connected. But there's two main ways of looking at it. That's the first time ever that I gave a talk and somebody knew that. That's good. Okay. So one way is the isoforms. You open a genome browser, that's what you usually get to see. Or if you're working in the splicing field, RNA splicing field like me, and you read any of the dozens of reviews or research articles, or hundreds or thousands of them, that what you'll see, alternative splicing events. So what you're seeing here are the green areas, all the alternative regions, and they can be either included or excluded. The dashed lines represent the splicing events that are splicing together the different segments. And if you just look at this, squint a bit, you'll see a huge difference between those two representations. One of them is very simple, very simplistic, and the other one is the isoforms. And of course, everybody, like the holy grail will be to understand all the isoforms in my samples, whatever I'm looking for, and what their exact abundance is. Of course, that's very hard, especially from RNA-seq data, which is noisy and sparse and biased, etc. And so the question is, can we could do something different? And so last year we came up with a different definition for how we think about splicing variations, and we called it local splicing variations. And the definition is rather simple and intuitive over splice graph. So let's look at a gene splice graph. The exons here are the blue rectangles, and the edges represent splicing events where you splice the exons together. When you look at the gene splice graph, every time you see a split in the graph coming out of an exon or into an exon, that would be a single source and a single target, okay? That would be a local splicing variation, or LSV. And it's local because there's always a reference exon you're coming out of or coming into. So that's a very simple definition, graph-based. But the nice thing about it is it corresponds directly to the biology, right? Because every time you see a split like this, it tells you something. It tells you that the spliceosome that is responsible for this has multiple choices, and it needs to decide which one of those it's going to splice together and in what ratios, okay? And the other thing, very nice thing about this is, unlike isoforms, which requires a lot of inference, hard inference, because we don't know the isoforms in a sample and the reads are very short, here we have direct experimental evidence to tell us what's the fraction of each one of those choices. And that comes in the form of junction spanning reads that span those junctions, okay? So the other advantage is, if you use this kind of definition, is it will capture all the previously classically defined alternative splicing events, okay? And notice that all those classically defined are very conveniently defined as binary events. You either go to this junction or you go to that junction. You skip the exon or you include it. But with this definition, you can find many more types of variations, including ones which we call as complex, that are seen here, which means they're going to have more than 2 junctions. And you can have multiple exons, multiple junctions, combinations of them, etc. Okay, so that's theoretically nice. Why would that be interesting? So before I jump into that, I'll just explain how we quantify splicing using these definitions. So here's a gene splice graph that is coming out of our software. It's called Magic, that I'm going to describe later. Every time you see a red junction, it means it was already annotated in the database of a transcriptome. Every time you see green, it means it's de novo. It's something that hasn't been annotated before. And so here is a cartoon. This is the exons. You can see here exon 8. Oops. And exon 8, this is the cartoon of exon 8 connected to the previous exons upstream of it. And this is what we call percent spliced in, or percent selected index. So the percent of the isoforms that include each one of those red or green or blue edges. And similarly, if this is in one sample, if I'm comparing 2 samples and I want to see differences, say between brain sample and a liver sample, I would be talking about delta psi, so changes of inclusion. So for example, the red guy over here, the red junction, the changes between the 2 conditions, tissue 1 and tissue 2, would be around 70% changes in inclusion. Okay, and this is usually what we care about in terms of splicing. What are the isoforms that are changing the inclusion level of those specific edges? Okay, so this is the basic of sort of what we're going to talk about today, and the differences between two conditions, which would be what usually people call differential splicing, okay, or DS. So the first thing to observe is if you start using this definition, if you just go to a database, you even just go to RefSeq in human, so you can't really go more conservative in a way than that, you're going to find that over 20% of splicing variations in human are complex. So they have more than 2 junctions, okay? If you start adding, if you go ensemble, you're gonna hit 33%. If you start adding RNA-seq data, multiple tissues, you're gonna have 30, 40% depending on what your reference that you're using. Similar numbers for mouse. So my point is, this definition is not something that is not there. It's already there. You can observe it even if you use very conservative definition. It's just hasn't been really defined before, okay? Now, of course, you can say, okay, it happens, but it's not really used, or it's not really meaningful. We've shown in that eLife paper that we published that those complex variations are actually highly conserved in the regulatory elements in the exon and the intron, far more than constitutive exons and far more than binary events, which everybody has been studying. Okay, and again, these are 30% of the variations in human. Okay. So measuring reproducibility. Let's say we buy into this and we want to do this, and we want to measure complex variations, binary variations. We want to measure them in the transcriptome. And I devise an algorithm, we did, to do this. Then the big question is, how do I actually show that my algorithm is doing what it's supposed to do, and how do I show that it's actually doing better than, say, other algorithms? That's hard, of course, because as we know, working in computational biology, it's very hard to have a golden standard, okay? Which is, you say, thousands of events that have been measured, and we know that's the value, et cetera, et cetera. So one way to look or think about it is what we came up here, which is a measure for reproducibility. And the logic is similar to what has been done for ChIP-seq, peaks, et cetera. It's just an adaptation of the same idea. And the idea is the following. I don't care what your measure, your score is. I don't care how you devise it, your probabilistic model. You're going to look at 2 conditions, and you're going to give me back a ranked list of all the things that you think are significantly changing between the 2 conditions. And now we're going to repeat the experiment. Okay, we're going to ask another mouse to contribute its liver and its brain, and we're going to do the same comparison again, and again rank them and see whether those high ranking are reproduced in that other set. Okay, so the x-axis here is how many events do we report, and the y-axis is how many of them are reproduced. And of course, when you start, the end is it's very fuzzy because the top ranking arm can jump around, and you have very little statistics, a number to get a stable statistic. And these are our algorithms and 2 versions of them, and these are the competition, other very usually used algorithms. Okay, and one of the additions in this last paper just got accepted in bioinformatics was the ability to handle an outlier. And I'm going to talk a bit about this later on. So one thing to consider is that even if you do this, right, there is no guarantees that what I'm actually showing you bears any resemblance to reality, okay? Why is that? I can give you a highly reproducible algorithm right now that's very simple, and it will be 100% reproducible. And it would go like this. You give me a list of genes. I will rank them alphabetically, okay? And when you give me another experiment, I'll rank them again alphabetically, and it will be very, very reproducible, right? But it has nothing to do with the signal, okay? So the point is, you can be reproducible, but highly biased by things that has nothing to do with the signal. Yes?
Single transition?
I mean the fact that I'm ranking, then I'm saying here I'm reproducing it, right?
No, I'm sorry, not the recent course. I mean just in this chart. It says fraction of events reproduced. Those events are the splice transitions?
Yeah, and so yes, and I didn't mention it, but one of the issues of comparing it, I didn't get into it, is all those algorithms, they speak different languages, right? One of them measures classical ones. The other one uses Salmon to compute isoforms and then looks for events. We're using this LSV language, et cetera. So one of the issues is how do you compare them? In a way, what I'm saying here is I don't care as much. Whatever language you want to use, I'm going to work with you. Okay, I just want to see how reproducible you are. You can define them any way you want, but I'll just want to see how reproducible you are. Okay, as long as you're not doing things that are, reporting the same thing twice, and you need to be a bit careful about it. But that's a great point. Okay. So what else can we do? So another measure is measure what we call putative false positives. And I call them putative because I can't prove that they're false positive. But the logic would be like this. If I'm comparing two groups, I'll compare two groups of things that are supposed to be identical, right? A bunch of livers and a bunch of livers from the same mouse and the same conditions, et cetera, et cetera. Biological replicates. Two groups of biological replicates. If my algorithm is working well, it will understand, right? If I compare two mice, of course, there is natural variability, right? If I'm comparing two groups, the algorithm should understand the variability here and the variability here. Basically, those groups should be very similar. So you shouldn't be reporting anything significant at large amounts, definitely compared to the comparison of, say, a brain and a liver, okay? So I can't prove that between the two groups they're false positive, therefore we're calling them putative false positive. Definitely, whoops, it's unfortunate, and now we're out. Okay, sorry about that. Okay. So what you see here is the number of reported events by each one of the methods, and this is the fraction of those reported number of events compared to the number that they report when they do compare two different groups, like a brain and a liver. So that's similar in spirit to what you would call a false discovery rate, but of course it doesn't follow the definitions of false discovery rates. Therefore, the reviewers called us on that, and we can't call it false discovery rate. But of course, it's highly related, and when you do simulations, you see the relations. But it's similar in essence of what you're trying to capture. Okay. And the other thing, of course, that you can do is you can do actual RT-PCR wet lab experiments to validate how accurate you are. You need to be very careful on how you do this. We also have a wet lab, so we can do that at large quantities. And we did this, and we measured correlations in R2 in terms of variance explained. And again, the algorithm is doing very well. So these are all ways that you can evaluate your algorithm. And maybe on your experiments and your data sets, our algorithm won't be as good, right? It's a function of the specific settings and what you're trying to do, but these are good to keep in mind in terms of way for you to evaluate whatever splicing algorithm you care about or you want to try on your data sets. Okay. Sorry, so what we do is we quantify those psi's, okay, from RNA-seq data, and then we do RT-PCRs, okay, which are low throughput. So you have a total of 50 of those in this case, which is quite a lot, okay, because you're doing it in two different conditions. That means 100, and you're doing it in triplicates, okay? So these are all different algorithms that are highly used in the community. SUPA uses Salmon to do isoform quantifications, and basically it looks at the splits and quantifies them. RMATS is an algorithm from Yixing's group, and Magic is our algorithm. And these are two flavors of it, but I don't have enough time to get into the details of it. Okay. So another possibly important take-home message, if you want to do this, is that de novo detection is really important. So many algorithms, what they do is they take a transcriptome and then they quantify splicing based on the annotation. What we've seen is, so our algorithm is able to take a known annotation, but then supplement it with everything else that it finds. And what we find is if we do this across mouse tissues, we increase the detection of more events that are significantly and with high confidence differentially spliced between the two conditions by 30%. This is what you're seeing here with de novo or non-de novo, but with the same reproducibility rate, okay? If you're comparing replicates or if you're doing RT-PCR. So this is not noise, and this is not something that is not reproducible. And so by assuming a transcriptome, you may be losing a lot of your signal, okay, depending on, again, on what you're looking. But these are normal tissues. And if you're looking at specific things that you really care about, if you're looking at variations introduced in cancer that goes crazy about something, or you're knocking down things, then you might very well missing things that are not in the annotation. So including de novo detection can be critical. Okay. Yep.
Does your model assume independent events at the different splice sites, or can they be correlated?
For variations?
Yes, when you're using your model to.
So each LSV is quantified separately based on the experimental evidence that comes out of the RNA-seq data. Yeah. You're talking about mechanistically, which is a very interesting question, but we're not going to get into it. Yeah. Okay. Dealing with outliers. So, it's Microsoft Research Center, and I thought I'll at least try to spend 2 minutes explaining a bit about something of how it works. Okay, so I'll do this about outliers. Let's see if I can get 2 minutes and give you an idea of how we think we can detect outlier and sort of control for them. So the idea is very simple. I get a batch, I get a replicates, but there might be a bad apple in the bushel, right? So there might be one that is an outlier, or two, or something like that. And the question is, how can I detect them? If you read the literature, they'll tell you, do PCA and look for outliers, which is great advice. I would recommend eyeballing the data. But how does that actually work? So I have a bunch of points here, and I have a point here, and then that's an outlier. Is that an outlier? Outlier? I mean, how? There's no real good way of defining it. And so intuitively, you would say outlier is something that is misbehaving, right? So how can you formalize this mathematically and look for those and control for those? And the other thing is, usually if you detect them, you just say, oh, throw it away. Right? You just wasted a lot of money and possibly statistical power, which is what we've shown in that paper. And so how do you do this? So the idea is very simple. We take all the LSVs that we find, and we say, okay, let's look for each LSV, right? We compute those psi's, and let's compute that in my group that are supposed to be bona fide good replicates. Let's compute the distance for each one of them, the maximal distance from the median in that group as a first pass estimator for how the group is supposed to look. And let's look at the distribution of distances that I see across all the LSVs. So this is a histogram. These are the distances, L1 in this case, and this is the probability mass for each one. And one of them you can see is an outlier. So what makes it an outlier, if you think of it, that will show up in your PCA or whatever, is that for a subset of the events, it will be misbehaving consistently, which is why you can call it an outlier. Because if it's misbehaving one, maybe it's just biological variability, and that's fine, right? So you can start calling it an outlier and detecting it if it's consistently associated with misbehavior compared to what you would expect. And so if you look at that plot of distribution, if you just cut it anywhere reasonable with high variability from the high distance from the median, say at 0.8, what you see is the outlier has an enrichment for this. And you can try different configurations. We did in the paper. Outliers will usually show up like this, okay? And so based on that, you can do a very simple thought experiment. You would say, if all of them are behaving nicely, okay, the number of things that are far away from my median should be roughly equally distributed, right? It's like an equal coin. So if I have t samples, it should be 1 over t, the chances, okay? In practice, of course, there's more variability, so you need a beta binomial for that. And it's going to be looking like this. So I can compute the weight that as a belief for me that this is an outlier, what do I observe in terms of the numbers that the coin landed on this guy compared to what I would expect from an equally likely distribution, okay? And that will give me a global weight for how much I think this replicate is indeed an outlier. And now the next thing I can do is I can look locally in a specific region and say, in that region, is there an enrichment? And I can score for each LSV how enriched is that region in that specific sample for being an outlier. And that would result in a local weight for each LSV. So the point I'm trying to make here is if you look at this distribution, these guys are probably bad, and I'm going to throw away that blue sample, but I'm going to lose all the other guys, all the money that I invested and all my possible statistical power with guys that are totally perfectly fine. So instead of throwing them, I can actually salvage them out by using this local weight, okay? And that's the gist of it. So hopefully that was clear enough. And what we've shown in the paper is you actually get better power and better accuracy with this. Okay, so I'll talk about how you can actually use this. So the algorithm Magique that I mentioned comes with a visualization package called Voila. And this is a glimpse of how the output looks. It comes in HTML5 with D3 interactive component. That means it lives in your browser. You don't need to install anything special. This is an LSV that you can see here, the quantification. It's connected to the genome browser or GTFs. And this is the splice graph that matches it, okay, for that specific gene. And you can see that we can handle a lot of complexity. I'm showing you things that were just generally, you wouldn't find in the literature anything that described those complexities because there was just only classical events that people were probing, okay? So if you want to focus in on them, you can remove, you can ask the package to simplify the view, and a lot of those edges that are very lowly expressed will remove. You can ask it to highlight that specific event. You can ask it to weight it, so you can easily see what are the used events, and you can click on it and go directly to the genome browser to see the LSV and all the raw reads. So you can go straight back to the raw reads as well. You can also hit that copy LSV button, paste it in a Galaxy server that we just released recently for this, and that will do all the, we will run an algorithm to do design for the primers to validate your events so you can do the validation, because those events become pretty complicated. And so basically we implemented an algorithm we use for our validations in the wet lab, so you can see all the isoforms that are associated with it. You'll see the protein domains that are affected, so you can start seeing what is the functional effect of those LSVs when you're changing them between the conditions. Okay, so this is all tools for you to use, assuming you're interested in variations in genes, in isoforms. Fast, vector accurate, so we care a lot about that, and people who follow Twitter have seen probably a lot of debates about who invented what by very prominent and extremely capable people. And you know, there is now a lot of emphasis of getting everything very efficiently and fast processed and executed, and we think it's important, but we also think about accuracy. So I like to show this kind of cartoon between, you know, kids that are arguing or discussing, and, you know, some say, I can compute really fast. The other says, so, okay, what's 123 multiplied by 65,000 something? And the other guy says, well, of course, it's 133, 6,780. He says, well, that's not even close. He says, yeah, but it's really, really fast, right? And so I show this as a joke, but I think, you know, especially depending on what you're trying to do, but it's important that we keep both in mind and focus on that, especially for RNA splicing when we're trying to understand what the hell is going on there. And so, to be fair, we also improved greatly Magique to make it able to process tons of data. So now it can handle GTEx, which we're working with. It can keep very low footprint of memory, so you can run it on your laptop, you can run it on an HPC, etc. And it's being updated, and the next generation is going to be faster and more lean and more capable. It's coming out soon. The next step is what we call Magique Hat, or Magic Hat, hat for heterogeneous. So the idea is that you can handle things like GTEx, and you can look for variations in them. And the idea is that you need a different statistical model. Because everything, if you think of it, everything that I told you about before is I had a bunch of replicates in liver, I had a bunch of replicates in brain, and I was comparing them. I'm assuming they're replicates, right? And then the next thing I told you is about outlier detection. So I had a bunch of replicates, but one of them was bad. So what I was trying to do, I was trying to salvage statistical power, but I was trying to detect who the outlier is, right? So I'm trying to control and remove the variability, because I'm assuming it's an outlier, okay? But if I have GTEx, these are not biological replicates. These are different people, different backgrounds, right? Many different confounding factors, and I have a ton of them. So I should have a different statistical method, statistical model. Instead of trying to repress the variability, I want to account for it, okay? And that requires, of course, efficiency, and it requires some different statistical model, and that's the things that are coming next. I'll just show you a brief view. Here is the same type of reproducibility, but now it's 45-degree angle, and this is if you use just expected psi or a t-test, even you're using currently Magique, or if you're using the new stuff in terms of the improvement in reproducibility, and these are the sort of variants around this, using GTEx samples. And how it works. The magic hat works basically like this. And, you know, we'll tell you later. So take-home messages, 25 minutes. Let's see what we've learned, hopefully. So there's many ways to think about, detect, and quantify and visualize variations, and that relates to the Sapir-Whorf hypothesis. That was the riddle I gave you, okay? So Sapir-Whorf hypothesis has nothing to do with math, okay? It has to do with linguistics. And the basic idea is that language determines thought, okay? So if you have certain constructs, that's what we use. So if we only talk about isoforms, when we only talk about AS classical events because that's how we were brought up when we read the literature, then we are never able to study other things, right? How can you study something if you can't even name it? So that's the basic concept, and that's why we think using this novel way, new way of, or terminology of looking at variations, and we've done that in that eLife paper and follow-ups, you can gain so much more insights. Accounting for complex variations is important. Hopefully I convinced you in that. De novo is also, can be critical. And I talked about how to assess differential splicing performance. You know, your mileage may vary. Whatever you decide to do, your data set might look different, and suddenly our algorithm doesn't perform well. So instead of taking my word that the algorithm is nice and great, check it out, but you know how you can assess those algorithms and different algorithms using these types of approaches. And I described some of our solutions, the Magique algorithm, the Voila visualization package, and the Magic spell. And the last thing I mentioned is how we want to scale up, and that relates to the previous talks, I guess, in terms of where the puck will be. Okay, and with that, I'll thank all the people that did all the hard work in my lab, and Kristen, who's a close collaborator and helps us make sure that all the wet lab experiments that we do are intact and they work very well. And the funding, and thank you for your time.
We have time for a few questions.
So one of the methods, I think it was Magique, it uses local weights to only discard regions from the replica. What if we have a zigzag-like distribution where certain adjacent regions would have very different local weights? Would it discard just few regions but keep the adjacent ones rather than larger stretches? And my second related question, how do you choose the length of the regions?
Right, so just to okay, so I think it's, possibly I didn't explain it very well. So the idea is that an outlier will have sort of an overabundance of things that, if you look at the histograms of cases that it doesn't agree very well, it will have an overabundance of those, right? And so what happens is if I look at a specific LSV, doesn't matter the location, has nothing to do with the location, okay? If I just look at that and I say at that level of disagreement, okay, from the median, or what we actually construct is based on the initial analysis, we construct of how a well-behaved, we're using those initial weights, those global weights, to say this is the histogram, okay? This is the histogram of how weights should be distributed on a well-behaved replica. Now we look at our case, okay, and we say this is your distance. In that amount of distance, just around it, that level of distance, I see how many do I land and how much do I expect, right? So if there is, I don't know, 8% over there, but I only expect to see 2%, the ratio between those will tell you those weights. And for each one of them, you do this. And how big is your window? Is that your question? So you can either sort of do it as an approach, so you try to minimize the distance. But what we usually do in terms of implementation, we just look at a small window around it, which is reasonable enough. Just a few percentage on that frequency. I don't remember the number, but it doesn't matter too much. Yeah.
Okay, so thanks for a nice talk. This is a really interesting topic in general, I think. So I actually have like a couple of questions that I'll try to state clearly. So just since you've spent so much time looking at these, one of my questions is, how important do you think slight variations in the targeting? So in other words, you're going from one junction and you go to the very end of the exon, or you go slightly into the exon. How often do you see those? How do you differentiate them? And do you have any sense of how biologically important that is, so the sort of partial exon inclusion or exclusion? And then the other question was, have you observed things that seem to be happening predominantly at the first or the last exon in annotated transcripts?
Okay.
Oh,
so
and the last one, sorry, is what do you think about long-read sequencing? Where does that fit into all this?
Right. So these are all great questions. So it's going to take us a bit of time to answer, to be fair. So small variations can affect, people have shown, not just us, other people have shown very nicely, like my postdoc advisor, Ben Blanco, have shown that you can affect, by including or excluding small parts, you can affect protein-protein interactions as one of the examples, and they've had a few very nice papers that illustrate this. We don't necessarily understand everything that happens in splicing, and definitely there's noise involved. I have a longer talk where I talk about also the noise component. Not everything is necessarily functional, but function is a big word. How do you define it? If I'm producing something which is not functioning properly instead of producing what I'm supposed to do, so this one doesn't have function, doesn't have as much as function, but its function is not to produce the other thing, right? And a good example of that is pretermination codon that leads to NMD. So if you do this, you're basically downregulating the expression of the gene, and that's a known case, and there's dozens of those cases specifically for splice factors that control their own level of expression like this. So that would be another example. Yeah, without taking too much time. So long reads. Long reads are great. We're definitely, I think that's part of the future of where this is going. Long reads right now, you don't get enough of them to quantify the actual isoforms in terms of quantifications. You can get indications that they exist. So I think a good future, and that's part of the project in the lab, is to combine those two, and some people have done that to some extent, and I think we can do more in that realm. Thank you.
I think we're gonna move to the next speaker. Thanks, Joseph. Let's thank Joseph again for the great talk.